Good afternoon. Today I am going to talk about fungi. In this presentation, I will talk about morphology and classification of fungi. The first group of microorganisms are bacteria that we have studied in detail. In case of bacteria, we studied morphology, classification, arrangement of bacterial cells, then various growth requirements. So in case of fungi, we are also going to discuss about its morphology as well as classification. What is fungi? Fungi are included into the kingdom of fungi, that is fungi kingdom. They are non-motile eukaryotic organisms. They may be unicellular or multicellular. They have rigid chitinous cell wall. Fungi are chemoheterotrophs and acquire the food by the process of absorption, and these fungi are reproduced with sexual or asexual processes. The study of fungi is called as mycology. Okay, so the fungi are non-motile, means they do not move from one place to another place like bacteria. The bacteria have the organ of locomotion that is called as flagella. This flagella is not present onto the fungal cells, and that's why these fungal cells are non-motile. These are eukaryotic microorganisms, or maybe simply we call as organisms, because microorganisms are called as those organisms which are of macroscopic size. Here some of the fungi or some of the type of fungi are visible to our eyes, which are not of microscopic size, but they are mainly included into the group of microorganisms. So they are eukaryotic. The difference between prokaryotic microorganisms and eukaryotic microorganisms is that the prokaryotic microorganisms do not have nucleus into their cell, while eukaryotic microorganisms or simply organisms have nucleus in their cell. Okay, so bacteria are prokaryotic while the fungi are eukaryotic. The fungi may be unicellular or multicellular. There are some of the group of fungi which are unicellular, means made up of single cell, while there are some of the fungi which are multicellular. They have rigid chitinous cell wall, means the cell wall of fungi is complex as compared to bacteria, and they contain chitin as well as cellulose into the cell wall. The cell wall is complex, the cell wall is tough as compared to bacterial cell wall. They are chemoheterotrophs, means they require organic compounds for energy as well as carbon. Okay, so this is the introduction about fungi. So over last 20 years, the incidence of serious fungal infections has been increasing. These infections are occurring as healthcare associated infections and in people with compromised immune system. In addition, thousands of fungal diseases affect economically important plants as well as the human beings also. But these fungi, means some of the fungi are beneficial. Okay, so in this presentation I am also going to talk what is the importance of fungi. Firstly, we will see the difference between fungi and bacteria. On the basis of various characteristics, the fungi as well as bacteria can be differentiated. Cell type. The fungi are eukaryotic while bacteria are prokaryotic. As we already discussed that fungi do not have nucleus. Fungi have nucleus in their cell, while bacteria do not have nucleus. Their genetic material is not surrounded by nuclear membrane. But in case of fungi, the genetic material, either DNA or RNA, is surrounded by nuclear membrane, and that's why they are called as eukaryotic microorganisms. Optimum pH, means at what pH these microorganisms grow well. So fungi has the optimum pH of about 4 to 6, while bacteria grow best at 6.5 to 7.5 pH, which is neutral pH. We can say that fungi grow at acidic pH. Okay. Then optimum temperature means the temperature at which the microorganisms grow well. So fungi has optimum growth temperature in between 25 to 36 degrees Celsius, while bacteria grow best at 32 to 37 degrees Celsius. So that's why after incubation of bacteria into the culture medium, generally 37 degrees Celsius temperature is chosen in case of in case of bacteria. But in order to store the fungi, the temperature is in between 25 to 36 degrees Celsius. Means less temperature or lower temperature is required for growth of fungi, and that's why the fungi are generally incubated into BOD incubator, that is biological oxygen demand incubator, which regulates the temperature itself. Means after increasing the temperature is automatically lowered, but that does not happen in case of general incubator, which is used for incubation of bacteria. So the optimum temperature for fungi is in between 25 to 36 degrees Celsius. Cell membrane. The sterols are present in case of fungi, while these are absent in case of bacteria. Sterols are mainly present into the cell membrane as well as cell wall of fungi. Then on the basis of oxygen requirement, they are strictly aerobic, while some of the fungi are facultative anaerobic. Strictly aerobic means which require oxygen. Oxygen is must for their growth as well as survival. And facultative anaerobic means this either may require oxygen or may not require oxygen. They may grow in presence or in absence of oxygen. These are facultative anaerobic. And the bacteria may be aerobic to anaerobic. On the basis of light requirement, means the fungi do not require the light for the growth, while bacteria means some of the photosynthetic groups require the light for their growth as well as survival. Then carbon source for fungi is organic, while for bacteria it may be inorganic or organic. Cell wall components of fungi are chitin and cell wall, while for bacteria it is peptidoglycan. So due to presence of chitin and cellulose, the cell wall of fungi is tough or rigid as compared to cell wall of bacteria. Then examples of fungal species are Penicillium species, then Candida albicans, Saccharomyces cerevisiae. Okay, these are the fungal species, and common bacterial species are Escherichia coli, Staphylococcus aureus, and Pseudomonas aeruginosa. So this is the general difference between fungi and bacteria. So there are also some of the common characteristics in case of fungi as well as bacteria also. The common characteristics are, these are microorganisms. These require culture medium for their growth. So for growth of fungi, the media containing high carbohydrate source, nitrogen source are required in the pH range of about 4 to 6 and in the temperature range of about 25 to 36 degrees Celsius. Media generally contain a source of carbon, nitrogen and vitamins. Glucose is the most widely utilizable carbon source and hence it is the most commonly used in growth media. Fructose and mannose are the next most commonly utilized sugars by fungi and are found in media from natural sources. Okay, so the most common natural occurring vitamin deficiencies are thiamine and biotin. So like bacteria also, fungi require various carbon sources, nitrogen sources, various energy sources, various vitamins, growth factors likewise. We will see the morphology of fungi. So the fungi can be classified on the basis of cell morphology. There are generally 4 types of fungi. These are molds, yeasts, yeast-like fungi and dimorphic fungi. We will see these classes one by one. First type is molds. Fungi which form mycelia are called molds or filamentous fungi. These mycelia means filaments, longer filaments occur during the growth of these molds. Okay, so these filaments are called as mycelia, and such fungi, such a kind of fungi are known as molds or filamentous fungi. The body, which is called thallus of a mold, consists of long filaments of cells joined together. These filaments are called hyphae. Okay, so these filaments are mainly made up of different cells, or these filaments are also called as the hyphae. In most of the molds, the hyphae contain cross walls called as septa, and in singular form it is called as septum, which divide into distinct uninucleate or multinucleate cell-like units. These hyphae are called as septate hyphae. Here into the images you can see it is a kind of mold. Mold is made up of hyphae. Okay, means these are the filaments. Hyphae are the cell units, and some of the hyphae may be uninucleate or multinucleate. Uninucleate means which contains single nucleus, while multinucleate means more than one nucleus are present into the hyphae. And these hyphae are mainly separated by a section that is called as the septum. Okay, and the hyphae which contain the septum are called as septate hyphae, and another type is non-septate hyphae, which do not have septum, which appear as long continuous cells with many nuclei, and these hyphae are called as non-septate hyphae or coinocytic hyphae. Into the image you can see, into the second image which are coinocytic hyphae, the septa are absent in such kind of hyphae, so that's why they are called as non-septate hyphae. Some of the septate hyphae contain multinucleate septate hyphae. Into the image that is not shown. Into the image only uninucleate septate hyphae have been shown, in which only single nucleus is present, but in case of multinucleate septate hyphae, number of nucleus are present within the hyphae. Okay, so this is the general morphology of these molds. The fungal species which are molds are Aspergillus niger and Penicillium notatum. The first antibiotic which was discovered is penicillin, and penicillin was produced by using the fungal species that is known as Penicillium notatum. Then next category is yeasts. Okay, before moving to the fungi, we will see in detail about generally these hyphae are about 2 to 10 micrometer in diameter. When environmental conditions are suitable, the hyphae grow and form a mass called as mycelium, which is visible to our naked eyes. Into the image you can see there is a braid. Onto the braid there is the growth of mold. You can see the braid is decomposed after production of this mold. Burshi we can say in Marathi. So that is also kind of a mold. And into the next image you can see when suitable nutrients are available for their growth, like bread contains number of nutrients, and that's why the fungi grow well onto the breads after their decomposition. Into the next image you can see the hyphae grow and form a mass called as mycelium, and these mycelia are visible to our naked eyes. The portion of mycelium concerned with obtaining nutrients is called as vegetative mycelium. The portion concerned with reproduction is called as reproductive mycelium, because it projects above the surface of medium on which the fungus is growing. Okay, like the fungus grows onto the bread, so we can see this fungus with our naked eyes. So likewise, molds decompose various breads, bakery products, as well as various foods. Okay, so instead of storing these breads or other foods into the refrigerator, the fungi grow, and the reason behind this is that the fungi grow at low temperature, while bacteria do not grow at lower temperature, and that's why into the refrigerator after some of the days the food decomposed due to production, due to growth of these molds. The next category or next type of fungi is yeasts. Yeasts are round, oval or elongated unicellular fungi. It is a class of fungi which is unicellular, while the molds are multicellular, you can see, and the yeasts are unicellular, which reproduce by the process of budding. Into the next image you can see it is the process of budding, in which the cell develops a protuberance which is called as bud, which enlarges and eventually separates from the parent cell. So into the first picture there is yeast cell on which the bud develops. Into the next step, bud fully develops, and which may form the chain of buds, and after some of the time the buds may be separated from the parent cell. So likewise, with the help of budding process, the multiplication of yeasts mainly takes place. So onto the culture medium, these yeasts form smooth creamy colonies, and the example is Saccharomyces cerevisiae. So here into the image you can see these yeasts are round or oval, which are unicellular cells, which can be differentiated from the molds. Then next category is yeast-like fungi. In some yeasts like Candida albicans, the bud remains attached to the mother cell and elongates, followed by repeated budding, forming chains of elongated cells known as pseudohyphae. These can be differentiated from true hyphae because they have a constriction at septa. The septa are also present at the branching point. On solid media, moist creamy colored colonies are produced. So here into the image you can see the bud remains attached after the reproduction. It does not separate out from the parent cell. So that's why it is called as yeast-like fungi. So fourth class of fungi is dimorphic fungi. Dimorphism means which exhibits two forms of growth, which may be temperature as well as CO2 dependent. So these fungi are generally pathogenic, which cause the diseases. They can either grow as mold or yeast. The mold-like forms produce vegetative and aerial mycelium. The yeast-like forms reproduce by budding. Frequently, this dimorphism is temperature as well as CO2 dependent. At 37 degrees Celsius, the fungus grows yeast-like, and at 25 degrees Celsius, it shows mold-like growth. For example, Histoplasma capsulatum. So that is the dimorphism regarding this. So these are fungi which are generally pathogenic, exhibit two kind of forms of growth, and that is called as dimorphism. So likewise, you can see into the image, these are some of the dimorphic fungal species, that is Blastomyces, Coccidioides, Himoncia, Histoplasma, mainly one. Into the images you can see when they are present, when they grow into the natural environment, they show mold-like growth, but at 37 degrees Celsius they show yeast-like growth. Likewise. So these are the four classes of fungi. Then what is the importance of fungi? Fungi are important sources of antibiotics. For example, penicillin, which is produced by using the fungal species, that is Penicillium notatum. Chrysopulvin, which is produced by using Penicillium chrysopulvum. Cephalosporin, that is Cephalosporium species. So most of the antibiotics are mainly produced with the help of these important fungal species. These fungi are naturally present into the environment. So these important fungi are isolated, their suitable strains are separated, and from them various antibiotics are produced. These antibiotics may be either antibacterial as well as antifungal antibiotics. So first antibiotic penicillin was produced with the help of Penicillium notatum. Nowadays Penicillium chrysogenum is one of the fungal species is used for the production of penicillin. Then these fungi are also the good sources of different enzymes, for example amylase, which is produced from Aspergillus species. Then molds are used into the production of citric acid, oxalic acid and gluconic acid. So different organic acids can be produced with the help of these molds. Fungi have been used to alter the texture, improve the flavor, increase the palatability and digestibility of natural or processed foods. For example, Penicillium species is used for repairing of various varieties of cheese. Okay, so cheese is mainly processed by addition of this fungi. Then edible means mushrooms. There are a wide variety of mushrooms you can see into the image. That is edible means which is eatable. These are the important sources of food. Okay. Then with the help of fermentation, the industrial alcohol as well as various beverages like wine can be produced with the help of these fungi. For example, Saccharomyces cerevisiae is used into the production of wine. Various bakery products are also produced. For example, baker's yeast is used for production of these bread as well as other bakery products. So fungi have capability to break down complex organic substrates. This is an essential activity into the recycling of carbon and other elements into the cycle of life, and that's why the fungi has given the importance into the nature as well as into the environment. So these fungi play two kind of roles. First role is beneficial and another one is harmful. Beneficial role means these are important sources of antibiotics, good sources of different enzymes used for production of various organic acids. These are also the food sources. Industrial alcohol and beverages are produced with the help of this fungi. That is beneficial role. But another category is harmful role. Means there are some of the fungal species, or we can say only few fungal species which causes the fungal infection. For example, Candida albicans, which causes the infections, and that infection is the fungal infection. Okay, which is also called as mycosis. So any fungal infection is called as mycosis. Mycosis are generally chronic, means long-lasting infections because fungi grow slowly. Mycosis are classified into five groups according to the degree of tissue involvement and mode of entry into the host. They may be systemic, subcutaneous, cutaneous, superficial or opportunistic. So these fungi have been used in biotechnology field also from many years. For example, Aspergillus niger has been used to produce citric acid for foods and beverages since 1994. Okay, the yeast Saccharomyces cerevisiae is used to make bread and wine. It is also genetically modified to produce a variety of proteins, including hepatitis B vaccine. Okay, Trichoderma is used commercially to produce the enzyme cellulase, which is used to remove plant cell walls to produce clear fruit juice. When the anti-cancer drug Taxol, which is produced by yew trees, was discovered, there was concern that yew forests of the US, okay, fungi are used as biological controls of pests, and in contrast to these beneficial effects, fungi can have undesirable effects for agriculture because of their nutritional adaptations, as most of us have observed that mold spoilage of fruits, grains, vegetables is relatively common, but bacterial spoilage of foods is not. There is a little moisture on the unbroken surfaces of such foods, and the interiors of fruits are too acidic for many bacteria to grow there. Jams and jellies also tend to be acidic and they have high osmotic pressure from sugars they contain. These factors also discourage bacterial growth but readily support the growth of molds. A paraffin layer on top of jar of homemade jelly helps deter mold growth because molds are aerobic and paraffin layer keeps out of oxygen. Okay. So this is the importance of fungi. So into the conclusion, I would say that the fungi are a group of microorganisms which are included into the kingdom fungi, which are non-motile eukaryotic microorganisms. They may be unicellular or multicellular. They have a cell wall which is rigid one, and the fungi, study of fungi is called as mycology. Fungi is differentiated from bacteria by various characteristics. Then there are 4 classes of fungi: molds, yeasts, yeast-like fungi, and dimorphic fungi. Yeasts such as brewer's yeast and molds such as Penicillium chrysogenum, which produces the antibiotic penicillium, are classified as fungi. Yeast cells tend to grow as single cells that reproduce asexually in a process known as budding, although a minority of species reproduce by fission. Many yeast species are capable of sexual reproduction and formation of the spores. In contrast, molds grow as masses of overlapping and interlinking hyphal filaments and reproduce by producing masses of spores in a variety of structures. This division between yeasts and molds based on growth morphology is not clear-cut, as some yeasts can produce hyphae under specific conditions, for example Candida albicans, while many normally filamentous fungi possess a yeast-like phase at some point in their life cycle. Fungi are eukaryotic organisms, that is, their cells possess a nuclear membrane. Consequently, there are many similarities between the biochemistry of fungal cells and human cells. Fungi are widely distributed in nature, occurring as part of normal flora of the body of warm-blooded animals, as decomposers of organic matter, and as animal and plant pathogens. Medically, fungi are an extremely important group of microbes, being responsible for a number of potentially fatal diseases in humans, but a significant number of fungi are of great benefit to humanity in terms of production of alcoholic beverages, bread, and antibiotic. Fungi have also been utilized for a range of molecular biological applications. Okay, so this is all about fungi. Thank you.